Welcome to the Science of Ageless Living. I'm Dr. Ethan Hausman Marquis, doctor scientist and the only peptide clinic physician in London with a published body of work specifically in this field. This is the Ageless Top 20 Peptide series. Every episode covers one compound, the real biochemistry, the honest evidence base, what we know, what we don't, and what the regulatory picture actually looks like. No hype, no discount codes, just the science. Today, SEMAX. SEMAX occupies a peculiar position in the peptide landscape. It is one of the most discussed cognitive compounds in online longevity and nootropic communities. It has a specific traceable origin story, not a supplement company's marketing department, but a government research institute. It has genuine peer-reviewed mechanistic data. It is in clinical use in one country for defined neurological indications, and it has almost no completed randomized controlled trial data of the quality that would satisfy a Western regulatory agency. That combination, real science, real use, real gaps, makes SEMAX one of the most interesting and most frequently misrepresented compounds in this series. The online discourse around it tends toward either uncritical enthusiasm from people who've used it and reported subjective cognitive effects, or blanket dismissal from those who note the absence of phase 3 trial data. Both responses miss what's actually interesting about this compound. So let's do it properly. To understand SEMAX, you have to understand where it came from, because the origin is not typical, and it shaped the kind of evidence that exists. In the 1980s, the Soviet Union had an active and well-funded program of neuropeptide research at the Institute of Molecular Genetics in Moscow, operating under the Soviet Academy of Sciences. The driving figure in this work was Nikolai Miasoedov. The group was working within a broader framework of understanding how peptide fragments derived from endogenous hormones could exert biological effects distinct from and sometimes more targeted than DCUE are the parent molecule. The starting point for SEMAX was ACTH, adrenocorticotropic hormone. ACTH is a 39 amino acid peptide produced by the anterior pituitary. Its primary job is to stimulate the adrenal cortex to produce cortisol. That much is standard endocrinology. But researchers had observed in the 1960s and 1970s, beginning with work by David de Weed in the Netherlands, that fragments of ACTH, particularly the ACTH 410 sequence, had effects on learning, memory, and behavior in animals that were entirely independent of the adrenal axis. You could administer these fragments to adrenalectomized animals with no adrenal glands, and still see the cognitive and behavioral effects. The behavioral activity resided in a portion of the ACTH sequence that had nothing to do with cortisol production. This was a significant observation. It suggested that the pituitary was producing peptides with direct central nervous system activity, that there was a class of neuropeptides operating in the brain independently of the peripheral hormonal cascade everyone had been focused on. Miasoedov's group took this further. They synthesized a series of peptide analogs based on the ACTH 4-7 sequence, the shortest fragment, that retained meaningful neuropeptide activity, and engineered modifications to improve stability and CNS penetration. The result was SEMAX, a synthetic heptapeptide whose core is the ACTH 4-7 sequence, methionine, glutamate, histidine, phenylalanine, with a proline glycine-proline tripeptide added at the C terminus. That C-terminal modification, proglypro, inzatzis is not arbitrary. It was specifically designed to increase resistance to enzymatic degradation and to improve the peptide's ability to penetrate the blood-brain barrier following intranasal administration. Proglypro is a fragment of collagen and appears in several naturally occurring neuropeptides. Its inclusion makes SEMAX substantially more stable and bioavailable via the nasal route than the parent ACTH 4-7 fragment would be. SEMAX was developed, refined, and eventually approved in Russia by the Russian equivalent of a regulatory agency for clinical use in the 1990s. The approved indications include ischemic stroke, transient ischemic attack, cognitive impairment associated with cerebrovascular disease, and peptic ulcer disease, that last one reflecting a separate line of investigation into SEMAX's effects on gastric mucosal protection. This regulatory history is worth holding in mind throughout this episode. SEMAX is not a gray market research chemical invented by a supplement company. It is a compound with a 40-year research history developed by a national research institute, approved by a national regulatory agency, and used in clinical practice. That doesn't make it fully validated by Western regulatory standards, and I'll be precise about what that distinction means, but it does place it in a different category than many compounds discussed in this space. SEMAX is a heptapeptide, 7 amino acids, molecular weight approximately 813 Daltons. That small size is relevant. It is substantially smaller than most therapeutic peptides, which contributes to its bioavailability across the nasal mucosa and potentially across the blood-brain barrier. It is administered intranasally in clinical practice as nasal drops rather than injection. This is important for understanding both its mechanism and its practical use. The olfactory epithelium offers a relatively direct route from the nasal mucosa to the olfactory bulb, and from there to the central nervous system. For peptides, this pathway can allow CNS delivery that bypasses the blood-brain barrier almost entirely via axonal transport and extracellular diffusion along olfactory nerve fibers. Intranasal delivery does not guarantee this pathway, and the proportion of intranasally administered peptide that actually reaches the CNS via this route versus systemic absorption from the nasal vasculature is a genuinely difficult pharmacokinetic question. But the nasal route for CNS targeted peptides is mechanistically rational, not just a convenience. What does SEMAX actually do at the receptor and signaling level? The primary mechanism that has attracted the most research attention, and that best explains the cognitive and neuroprotective effects seen in animal models, involves brain-derived neurotrophic factor, or BDNF, and its receptor, TRKB. BDNF is arguably the most important growth factor for neuronal health, plasticity, and survival. It promotes synaptic strengthening, supports neuronal differentiation and survival, and is central to the cellular processes underlying learning and memory consolidation, particularly long-term potentiation in the hippocampus. BDNF levels decline with age, with chronic stress, and with a range of neurological conditions. Low BDNF is a consistently replicated finding in depression, cognitive decline, and neurodegenerative disease. Increasing BDNF, or upregulating TRKB signaling, is one of the most discussed targets in neuropharmacology. Multiple studies, predominantly in rodent models, have shown that SEMAX administration increases BDNF expression in the hippocampus and frontal cortex. The magnitude of this effect varies across studies, as does the time course, but the directional finding, SEMAX upregulates BDNF, has been replicated in several independent laboratories. The upstream mechanism by which SEMAX drives BDNF expression is not fully resolved. Some evidence points to activation of melanocortin receptors, specifically MC4R and MC5R, which is consistent with SEMAX's structural derivation from ACTH, since ACTH and its fragments are natural melanocortin receptor ligands. Melanocortin receptor activation has established links to downstream signaling pathways, including CAMP, PKA, and MAP-K-CHOS ERK cascades that can drive transcription factor activation and ultimately BDNF gene expression. But this is a plausible mechanistic chain, not a fully validated one. The precise receptor to BDNF pathway for SEMAX specifically has not been mapped with the resolution you would want for a full mechanistic claim. A second mechanism with good supporting evidence involves serotonin transporter regulation. Studies have shown that SEMAX modulates the serotonin transporter, SRT, in brain regions including the hippocampus and prefrontal cortex, effectively increasing synaptic serotonin availability. This effect is in the same directional space as selective serotonin reuptake inhibitors, though the mechanism is different and the magnitude is not comparable to therapeutic doses of antidepressants. The serotonergic effect may contribute to SEMAX's observed effects on mood and anxiety in some animal models, though extrapolating from rodent serotonin pharmacology to human mood effects requires significant caution. Third, and particularly relevant to the stroke and neuroprotection indications for which SEMAX is approved in Russia, there is evidence that SEMAX reduces neuroinflammation. Specifically, studies in ischemia models have shown SEMAX attenuates microglial activation and reduces pro-inflammatory cytokine expression in paralesional brain tissue following ischemic injury. The proposed mechanism involves modulation of NF-κB signaling, the same transcription factor we discussed in the context of TLR9 and TA1, but in this context, operating in neuroinflammatory rather than peripheral immune pathways, reducing neuroinflammation in the acute and subacute post-ischemic period is a rational target for neuroprotection, and SEMAX's effects in ischemia models are among the more compelling data in this field. A fourth mechanism worth noting involves the hypothalamic pituitary adrenal axis, the HPA axis. Given that SEMAX is derived from ACTH, the question of whether it affects cortisol production is not trivial. The evidence here is actually reassuring for most use cases. SEMAX, specifically the ACTH47 derived core sequence, does not appear to meaningfully activate ACTH's primary adrenal receptor, MC2R, at the doses used. The behavioral and neuropeptide activity fragments from ACTH had already been shown by DeWeed's group to be dissociable from the adrenal stimulating activity. SEMAX, as engineered, appears to retain this dissociation, but I want to flag this as an area where the absence of systematic human endocrine monitoring data in the published literature is a genuine gap rather than confirmed safety. Those are different things. Now the evidence. And here I need to be direct. The evidence for SEMAX is thinner than for thymosin alpha-1, thinner than for some other compounds in this series, and structured in a way that reflects its origin, a Russian government research program, rather than the Western Phase 2/3 randomized trial framework. What that means in practice is this. There is a substantial body of preclinical data, animal studies, cell culture work, ex vivo models. Ezio Nausaucere is Simordi Imelti was logically credible and published in peer-reviewed journals, including international journals. There is a smaller body of human clinical data, some of which is published and some of which exists in Russian language gray literature that is difficult to access and has not been replicated or independently assessed by Western research groups. And there is essentially no randomized controlled trial data that would meet the evidentiary standards of the FDA or EMA for a new drug application. Let me take you through what actually exists, domain by domain: ischemic stroke and cerebrovascular neuroprotection. This is the indication with the most human data because it is the primary approved indication in Russia. The preclinical foundation is strong. Rodent and rat models of middle cerebral artery occlusion, the standard experimental ischemic stroke model, have consistently shown that SEMAX administration reduces infarct volume, improves behavioral recovery, and attenuates inflammatory markers in paralesional tissue. These effects have been shown both when SEMAX is given prophylactically and when given in the acute post-ischemic period, with the post-ischemic data being more clinically relevant. The human data comes primarily from Russian clinical trials conducted in the 1990s and 2000s, examining SEMAX administered intranasally in patients with acute ischemic stroke and with post-stroke cognitive impairment. These trials reported improvements in neurological deficit scores and cognitive outcomes compared to standard care alone. The studies were published, many in Russian-language journals, some in international publications. The design of these trials, however, does not uniformly meet modern, randomized controlled trial standards. Blinding was inconsistent, outcome measures varied, and independent replication by groups outside Russia has not occurred. This is not the same as saying the data is fabricated or meaningless. It is saying that the evidentiary weight is substantially below what would be required for a Western regulatory submission, and therefore below what should be required for confident clinical claims. Cognitive function in healthy subjects and mild cognitive impairment. This is the area of most direct relevance to the longevity and nootropic use case, and it is the area where the evidence is most limited. There are animal studies, particularly in aged rodents, showing that SEMAX improves performance on learning and memory tasks, with associated increases in hippocampal BDNF. These are consistent with the BDNF-TRKB mechanism and are interesting preclinical observations, but aged rodent cognition data extrapolates poorly to human cognition, and the mechanism of cognitive aging in rodents does not map cleanly onto human cognitive decline. In humans, the data on SEMAX for cognition outside the context of established brain injury is thin to the point of being essentially absent from the peer-reviewed literature that would satisfy a systematic reviewer. There are case series, observational reports, and a substantial body of subjective user reports. But these are not evidence of efficacy in the scientific sense. They are signals that deserve formal investigation. The honest position on SEMAX for cognitive enhancement in healthy adults is this: the mechanistic rationale is coherent. Upregulating BDNF in hippocampal circuits is a plausible path to improve memory consolidation, but the human evidence supporting that extrapolation does not yet exist in an evaluable form. Stress, neuroprotection, and anxiety. There is a line of animal research showing that SEMAX reduces anxiety-related behavior and attenuates the neurobiological effects of chronic stress, including stress-induced reductions in hippocampal BDNF. The serotonergic mechanism is the most plausible explanation for these findings. Again, interesting preclinical observations, not human evidence. Optic nerve regeneration. This is a distinct and somewhat unexpected line of research that has attracted attention in Russian ophthalmology. Studies conducted at the Helmholtz Moscow Research Institute of Eye Diseases examine SEMAX as an adjunct treatment in patients with optic nerve atrophy, a condition of progressive optic nerve degeneration for which treatment options are limited. Reports from these studies describe improvements in visual acuity and visual field parameters in SEMAX-treated patients compared to controls. The data here shares the same quality limitations as the stroke data: small trials, variable methodology, limited independent replication. But it is distinctive because the theoretical basis, BDNF-mediated neuroprotection of retinal ganglion cells, is consistent with what we know about BDNF in the visual system. Safety data. The safety record for SEMAX across the Russian clinical literature and from intranasal use at therapeutic doses appears clean. The most common reports are mild local irritation at the nasal mucosa, entirely expected with any intranasal peptide formulation. Systemic adverse events are not a prominent feature of the published literature, and the absence of adrenal activation at therapeutic doses is consistent with the pharmacology. There are no documented serious adverse event signals from the clinical use data available. But the caveat here is the same as above. The absence of a safety signal in a literature that is not comprehensively accessible and has not been subjected to full systematic review is not the same as a fully characterized safety profile by Western regulatory standards. Let me now address the elephant in the room, because anyone listening to this who has spent time in the SEMAX online community will already be thinking about it. The subjective user experience reports. SEMAX has generated an unusually consistent pattern of subjective cognitive reports across independent user communities. People who use it describe increased verbal fluency, improved working memory, heightened ability to sustain focus, and, in many accounts, a quality of mental clarity that is perceived as distinct from stimulant effects. Some describe anxiolytic properties. The reports are widespread, they span different demographics and use contexts, and the consistency of description across people who have no contact with each other is notable. I want to be precise about what this means and what it doesn't. Consistent self-reported effects from a pharmacologically active compound with a coherent mechanism are not nothing. They are a signal worth attending to. The pattern of reports is consistent with what you might expect from a compound that upregulates BDNF signaling and modulates serotonin availability in prefrontal and hippocampal circuits. The subjective experience of people using SEMAX is not incoherent with the pharmacology, but subjective reports, however consistent, are not evidence of efficacy in the scientific sense. Placebo effects in cognitive domains are substantial, some of the largest in pharmacology. Expectation effects, particularly in communities that have developed strong positive narratives around a compound, can generate consistent reports of benefit that may have little to do with the compound's actual pharmacological effects. The absence of a randomized placebo-controlled trial means we cannot distinguish between a genuine pharmacological effect and a highly consistent expectation-driven response. What the subjective reports tell us is that SEMAX is worth investigating properly. They are not a substitute for that investigation. SEMAX's regulatory position is straightforward and in many ways more clearly defined than some compounds in this series. In Russia, it is a registered pharmaceutical product approved for clinical use, available by prescription, and manufactured to pharmaceutical standards under the trade names SEMAX and its variants. It is not a gray market compound in Russia. It is a licensed drug. In the United Kingdom and the European Union, SEMAX does not hold marketing authorization. It is not a licensed medicine. It is not on the controlled substance schedule. It can theoretically be imported for personal use under the personal import rules or prescribed by a physician as a specials product, an unlicensed medicine, under their professional discretion. But it has not gone through EMA review, and no UK or EU physician is prescribing it within a formally approved indication. In the United States, the situation is similar. SEMAX is not FDA approved. It is not a controlled substance. Its legal status for import and possession is ambiguous, as it is with most unapproved peptides, and it sits in the same regulatory gray space as many other compounds discussed in this series. The quality control issue is worth flagging explicitly. Because SEMAX is not manufactured under Western GMP regulation for the market in which most listeners are purchasing it, the compound available through peptide research suppliers varies in purity, authenticity, and concentration. This is not hypothetical. Independent testing of peptides available in the research chemical market has consistently shown variability. Someone who reports using SEMAX and experiencing strong effects or no effects may be partly reporting a quality and purity variable rather than purely a pharmacological one. This is a material clinical risk that does not apply to the Russian pharmaceutical product used in the clinical studies. Here is where SEMAX sits in the longevity context specifically. The longevity rationale for SEMAX is primarily neurological, maintaining cognitive function, supporting neuroplasticity, and potentially offering neuroprotection against age-related decline in hippocampal circuits. The BDNF framework makes this mechanistically coherent. BDNF declines with age. Hippocampal neurogenesis and synaptic plasticity decline with age. Memory consolidation, particularly episodic memory, is one of the earlier and more consistent cognitive domains to show age-related decline. A compound that robustly upregulates hippocampal BDNF signaling would, mechanistically, be a rational candidate for neuroprotective and pro-cognitive effects in an aging brain. The problem is the distance between that mechanistic rationale and demonstrated human efficacy. BDNF is a compelling drug target. It has been compelling for 3 decades, and the history of interventions that raised BDNF in animal models without translating to human cognitive benefit is a long one. Most antidepressants increase BDNF. The relationship between BDNF elevation and cognitive benefit in humans is far more complicated than the rodent literature would suggest. Exercise, to use the most evidence-based comparator, robustly increases hippocampal BDNF in humans, is associated with measurable improvements in hippocampal volume and episodic memory, and has a body of randomized trial evidence supporting cognitive benefit in aging adults. The exercise BDNF cognition pathway in humans is probably the best validated version of the framework that SEMAX is pharmacologically engaging. Whether a peptide that elevates BDNF through a different mechanism achieves comparable or additive effects in humans is genuinely unknown. A second longevity consideration is the neuroinflammation angle. Neuroinflammation, chronic low-grade inflammatory activation in the central nervous system, driven by microglial senescence and peripheral immune infiltration, is increasingly recognized as a driver of neurodegeneration and cognitive decline in aging. SEMAX's attenuation of neuroinflammatory markers in ischemia models is interesting in this context. Whether it has meaningful neuroinflammatory effects in the non-injured, chronically aging brain is a separate question, and not one the current data answers. The honest gaps for SEMAX are substantial, and I want to be as precise about them as I was about the evidence. There are no completed randomized controlled trials in healthy aging adults with cognitive or longevity-relevant endpoints. The evidence base for the use case that most people in this community are actually interested in, sharper cognition in a healthy 45 or 55-year-old, is essentially absent from the peer-reviewed literature. The optimal dose, frequency, duration, and route of administration for any longevity application are entirely undefined. The Russian clinical literature uses doses of 300 to 900 micrograms intranasally per day in stroke patients. Whether this translates to the healthy adult context and whether it represents the right dose for a BDNF-driven neuroprotective effect versus a more acute injury repair context is unknown. The durability of any effect is not characterized. It is not known whether repeated SEMAX administration produces tolerance, desensitization of the receptors through which it acts, or whether effects are maintained across extended protocols. This is not a trivial question for a compound proposed for chronic use in a longevity context. The pharmacokinetics of intranasal SEMAX in humans. What proportion actually reaches the CNS via the olfactory route? What systemic bioavailability looks like? What the half-life is, are not fully published in accessible literature. These are fundamental pharmacokinetic questions that you would expect to be answered before making confident clinical claims about a compound, and they are not answered for SEMAX in a way that is fully evaluable from the published data. And the long-term safety profile in repeated use outside of the acute clinical contexts studied in Russia is not established. The compound appears safe at the doses and durations studied. Extended use in healthy adults over months or years is a different safety question. Here is where SEMAX sits in 2026. It is a synthetic heptapeptide derived from ACTH 4/7, engineered for enhanced stability and intranasal bioavailability, with its primary mechanistic action centering on BDNF upregulation via melanocortin receptor-mediated signaling, secondary serotonergic modulation, and attenuation of neuroinflammatory cascades. It has a credible origin in serious government-funded neuropeptide research. It has a substantial body of preclinical evidence that is mechanistically coherent and directionally consistent. It has human clinical data in ischemic stroke and related cerebrovascular conditions, but that data is structurally limited by the design and accessibility constraints of the Russian research context and has not been independently replicated to Western phase 3 standards. For the cognitive longevity application that most people using it are interested in, the human evidence does not yet exist in a valuable form. The mechanistic rationale is coherent. The preclinical signals are interesting. The subjective user experience data is consistent but confounded. The human trial evidence is absent. What I will say is this. Among the compounds in this series, with a primarily cognitive rationale, SEMAX has one of the more coherent mechanistic stories and one of the better established safety profiles, even accounting for the data quality limitations. The gap between the evidence that exists and the evidence that would be needed to make confident clinical recommendations is substantial. But the gap is a research gap, not a red flag. That distinction matters. If you are a researcher or a physician looking at the landscape of under-investigated cognitive compounds with legitimate mechanistic foundations, SEMAX is on the list. If you're a patient being offered SEMAX at a clinic with confident claims about cognitive enhancement, you should ask your prescriber to walk you through exactly which human randomized controlled trials support those claims. And to be honest about the answer, if this episode has been useful, share it with someone navigating this space, particularly anyone being offered cognitive peptide protocols without a serious conversation about the difference between mechanistic plausibility and clinical evidence. This field deserves more precision and less noise. In the next episode, we'll be covering BPC 157, the most widely used peptide in the longevity and performance clinic world, and perhaps the compound where the gap between the evidence people believe exists and the evidence that actually exists is largest. We'll do it the same way. Mechanism, evidence, honest gaps, no shortcuts. I'm Dr. Ethan Hausman Marquis. This is the Science of Ageless Living.